This is Ville Kaila, a biochemist. He's working on a fundamental mystery. How can the cells in our bodies be charged with energy so that we have the power to move, to live? No one knows exactly how it works, but Ville is investigating special proteins within our cells which capture and convert energy from the food we eat, turning it into an electrical charge so that our cells become like charged batteries that power us.
In our cells, we have this wonderful membrane-bound machinery specialized in capturing energy and converting it into an electrical gradient across the biological membrane. And it's this flow of charge that creates basically the universal currency that powers everything in our cells.
To understand this, Ville must cross subject boundaries. His research includes biology and chemistry and quantum physics.
So we study these questions by combining both techniques and information across various disciplines, particularly chemistry, physics, and biology. And nature doesn't care really what we call these, whether it's chemistry or physics.
But before he began seriously studying biochemistry, Ville's career goal was to be a violinist. He studied music at the prestigious Sibelius Academy in Finland, though he also had a great interest in science.
At one point, I had to make the difficult choice of basically choosing between becoming a scientist or a musician. And this was a very tough decision, but it was very important for me to be able to do science at the highest possible level.
Once the decision was made, things moved quickly. Ville had already earned his PhD at the age of 24, followed by about 10 years of research in the US and Germany, among other places. But a while ago, he came back to the Nordic countries now as a research scientist at Stockholm University.
Now this is the tricky part, getting the bubble out.
And his entire team packed their bags and came here with him.
So 2019, when we moved from Munich to Stockholm, I was very fortunate that my research team wanted to move with me here. This was, of course, great that they wanted to do this, but also very efficient because we could continue the research right away here in Stockholm.
What Ville and his team now want to understand is one of the most fundamental functions of all living things: how cells absorb and convert energy. But just because it's basic doesn't mean it's easy. Far from it.
It's really fascinating how life is basically based on moving elementary particles, protons and electrons across very large distances.
Certain proteins in the cell membrane are specialists in capturing energy, for example, from the food we eat, and converting it into an electrical charge within the membrane. If you zoom in even closer, you'll see that at the smallest level, these proteins move atomic particles, electrons and protons. How this works in detail is the mystery that Ville is working to solve, in order to understand exactly what happens when particles at the atomic level are moved and cause the cell to become electrically charged, just like a small battery.
In order to get a structural picture of these proteins, we use this technique called cryo-electron microscopy. And in this method, we freeze our proteins in very cold temperatures, around -200 centigrades. And then the protein complexes are frozen both in time and space. And then by shooting these proteins with electrons, we can collect reflections and get insight into the three-dimensional shape of these protein complexes.
Ville likens the technique to this series of images taken in 1878 by photographer Edward Muybridge. For the first time ever, Muybridge managed to prove that a galloping horse momentarily has all of its hooves off the ground. This had long been suspected, but until the right technology was in place, in this case a camera, it had been impossible to prove.
So like this galloping horse, we do it a little bit in a similar way to get a detailed insight into the protein during its action by combining various techniques.
To understand the whole process, Ville and his team have to make mathematical calculations, as well as using both classical physics and quantum physics. With the help of a supercomputer, they can then predict how the elementary particles will move in the cell. They then run tests on sort of mini-cells that they've created themselves in the lab, where they can be compared with the theoretical calculations.
We have seen that many mutations in these protein complexes have been linked to various serious diseases, from mitochondrial disease to even Parkinson and Alzheimer. And it's fascinating that this information could basically provide a basis for possibly creating new drug molecules against these severe diseases.
Studying the machinery of life can also be inspiration for more tangible technological advances. Engines and machines that we humans have built are often quite inefficient when it comes to energy conversion. Whereas in biological systems, like the proteins in our cells, the efficiency rate is almost 100%, much higher than anything we've been able to manufacture so far.
Learning how nature creates these highly efficient charge transfer processes allows us to provide new information that could contribute, for example, for sustainable energy technology. It's really remarkable to learn about this wonderful membrane-bound machinery, this micro-universe of our cells and how they actually operate.